Good morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to today's webinar titled Understanding Advanced Cardiac Tissue Slice Applications, featuring Dr. Bradley Palmer and Dr. Matthew Caparizzo, both of who are assistant professors of molecular physiology and biophysics at the University of Vermont, and Dr. Benjamin Lee, a fellow of cardiovascular medicine with the University of Pennsylvania. Together, the three of them will provide a better understanding of how cardiac slices and the IonOptix Cardiac Slice System can be used to both complement and improve traditional cardiac investigations. All right, so now I'm very pleased to welcome our first speaker for today, Brad, to join me to kick us off.
All right. Welcome to today's webinar. I'd like to present to you our use of an experimental model system called the cardiac slice and demonstrate to you that the cardiac slice can be used to mimic the cardiac cycle and in particular the Frank-Starling law of the heart. I show this slide to remind us that we're focused on the function of the left ventricle and that blood flows through the left side of the heart from the left atrium into the left ventricle and from the left ventricle into the aortic root. The mitral valve and the aortic valve are very important as they direct blood flow in that order and prevent any retrograde flow. Now on the left side of this slide, you'll see a functional MRI image of a human heart. And this image shows the chambers of the left atrium and the left ventricle throughout the cardiac cycle. And the right side of the slide shows blood volume in the left ventricular chamber. And we see that immediately after initiation of a contraction, there is no change in ventricular volume for a short time before the aortic valve opens. This first phase of the cardiac cycle is called isovolumic contraction. And then, as soon as the aortic valve opens, blood is ejected until the aortic valve closes again. And isovolumic relaxation follows until the mitral valve opens, which allows the chamber to fill with blood. And now I'm showing you the complete Wiggers diagram with the left ventricular pressure throughout the cardiac cycle. It is the relationship between left ventricular pressure and left ventricular volume that we can mimic in vitro using a cardiac slice. And I'll show you how we do that, but first, let's take a closer look at the pressure-volume relationship. The relationship between pressure and volume is often visualized by this phase diagram where the pressure is plotted against volume, and the four phases of the cardiac cycle can be seen as the sides of a closed loop. And this loop is called a work loop because the area within the loop defines the mechanical work produced by the pumping action of the ventricle. Now, there are two important landmarks of this work loop I'd like to point out. The first landmark is end diastolic volume, shown with an orange dot. And this is also called the preload and is the chamber volume immediately before contraction. The second landmark is noted when the aortic valve opens and shown here with the blue dot. And this is also called the afterload and refers to the pressure in the aorta that the left ventricle must eject against. So now we can get to the Frank-Starling law of the heart, which can be described as this. With increasing filling of the left ventricle, the left ventricle will generate more pressure and eject more blood. And because there is more blood ejected, the afterload is typically slightly elevated, and the extent of contraction is slightly reduced, at least compared to the prior preload condition. And the slope of the pressure volume measurements at end systole is called the end systolic pressure volume relationship and is often used to indicate the intrinsic contractility of the heart. The increase in pressure development with increasing pressure, The increase in pressure development with increasing preload is known as the Frank-Starling law of the heart, named after Otto Frank and Ernest Starling, who described this phenomenon over 100 years ago. The underlying mechanism that causes this response is still not completely understood, but as a measure of intrinsic contractility, it is very important, and we can mimic this phenomenon using the cardiac slice. So here's a good place for me to learn about the experiments that you wish to perform. So Sydney, could you please start the poll?
You got it, Brad. So
Oh, I'm
polls
so sorry.
launched. The question is, when you work with cardiac muscle, are you concerned about monitoring diastolic sarcomere length? Yes or no? Back to you, Brad.
Thank you very much. So the cardiac slice is a slice of ventricle between 100 to 300 microns thick. And the slice is cut down to a few millimeters long and a few millimeters wide and placed on an apparatus, as you see here in the bottom right of this slide, that allows for control of its length while also monitoring contractile force. Now, if you're interested in more details about preparing a cardiac slice, please check out the Handouts tab, where you can download a list of references as well as several links to instructional videos. There are two important points I would like to make about the system. First, the apparatus monitors contractile force produced by the slice. And the length is controlled using a force feedback system so that the work loop can be generated. And if the cardiac slice is thin enough, sarcomeres are visible through an inverted microscope and can be used to establish diastolic sarcomere length. And I'll show you an example of collection of these data. So here you see a data collection interface for the three measurements. The top trace monitors motor position and indicates muscle length. The middle trace shows muscle force, and the bottom trace shows sarcomere length. And if you look closely, you can see sarcomeres in the video window just below the bottom trace. So I'll start the video here. And you'll see that sarcomere length measurement is stable in diastole. And when I start the force feedback control of the motor, the muscle is shortened at the correct time to generate a work loop. And you'll see it starting right about here. So the recorded work loop looks like this. And recall that this is a force length work loop. The recorded force and length are shown to the right, and the preload and afterload are indicated here with the orange dot and the blue dot. Notice that in this system, the preload is set by force feedback and therefore represents end diastolic force. These traces mimic the pressure volume work loop of the left ventricle, and with a prescribed preload and afterload. But we can also collect data with a change in afterload. So I'm going to show you another video here where you'll see the data collected over different afterloads. And instead of sarcomere length, you'll see afterload in the bottom trace. And you'll also see the work loops generated in real time. So you see the work loops being generated right now at the initial afterload, but using different afterloads, we'll get a change in the amount of contraction that occurs. So there's their first change in afterload. Here's our next change in afterload, and you'll see there's less shortening with increasing afterload. And there's our last afterload. But using these different afterloads allows us to later calculate the end systolic force length relationship. So I've summarized the results here. Where you can see that with increasing afterload, the peak force becomes higher and the extent of contraction is reduced. In a similar way, the preload can be changed to engage the Frank-Starling response. And you can see here with this in this panel that with an increase in sarcomere length shown in blue, there is an increase in developed peak isometric force. And then finally, in the bottom right panel, we can show the force length relationship for the various preloads and various afterloads. We typically normalize these measures to muscle length and cross-sectional area to relate stress and strain. And we can calculate the end systolic stress-strain relationship, which indicates the intrinsic contractility of the cardiac slice. So in conclusion, I'd just like to note that cardiac slices can be used to mimic the pressure-volume relationship of the whole heart while allowing continuous monitoring of sarcomere length. And with increasing sarcomere length, the cardiac slice demonstrates the Frank-Starling law of the heart. Now, Sydney, I was wondering if you could please tell me the results of the poll.
Absolutely, Brad. So our first poll, when you work with cardiac muscle, you're concerned about monitoring diastolic sarcomere length. We had 67% of our audience say yes, and 33% said no.
All right.
And for our second poll, what species is best represented by your work with cardiac muscle? We've got rat in the lead at 40%, mouse second at 30%, human next at 22%, other at 8%, and rabbit is only 1%.
All right, that's all good to know. I would like to note that the sarcomere length, as we've seen here, the sarcomere length is an important determinant of cardiac function. And for many studies, but certainly not all, the sarcomere length will be important to monitor or to set when you perform your experiments. So in conclusion, or I'd like to at least acknowledge the Terracciano Laboratory at Imperial College London. Cesare Terracciano and Fotis Patolis were very instrumental in teaching me how to generate cardiac slices. And I'd also like to thank my colleague, Matt Caparizzo, and his laboratory members here at the University of Vermont. And with that, I'll let Matt take over.
And okay, so I'm Matt Caparizzo. Thank you guys for the opportunity to share the work with you. Brad gave us a nice introduction to systolic function and the Frank-Starling mechanism. My research really focuses on sort of the other end of that. So we're interested in how the heart muscle relaxes and how this improves cardiac output by improving diastolic function. And the SLICE platform has been really instrumental in helping us to look at the effect of colchicine there. Sorry, this is messing my thing up. Okay, so I think that most of us are probably familiar with the purpose of the heart is to pump blood through your systemic circulatory system, deliver oxygen and nutrients. Your heart is a pump, but it's also a muscle, and it's powered by these cardiomyocytes here. I'm just trying to get my laser pointer off. Okay, which don't divide. They're about 100 microns long, and they're these rod-shaped cells, and they're programmed to live as long as you do. So if you live to be 100, these cells are going to beat over 3 billion times. And so this is a common technique in the cardiovascular field to isolate these cells. You can image the calcium and see calcium is inducing contraction. These cells is sort of a metric for systolic function or cardiac contraction and relaxation in a petri dish. And these cells are what's responsible for enabling your heart to respond to all the different stresses that you have as the day goes on. So hopefully we're all sitting here at rest. Our hearts are pumping about 5 liters a minute, but a trained athlete during exercise can pump up to 35 liters a minute. So this is a huge change that can occur in a short period of time. And it's these cells that are responsible for this adaptation to stress. So when your muscle cells work harder, I think we're also familiar with what occurs, right? They hypertrophy. And so here's an example of Arnold Schwarzenegger suffering from skeletal muscle hypertrophy. And of course, we don't look at Arnold and go, wow, this person is so remarkably unhealthy. Quite the contrary. And sort of the same thing is true for your heart, and that your heart, in response to a stress, so these stresses can be positive, exercise, for example, or they can be pathological, hypertension, diabetes, and obesity. You get hypertrophy of the heart. And so these are some examples of human hearts that we received in the Ken Margulies lab. And so a healthy heart here, and roughly on the same scale, a hypertrophied heart. Looking at this heart, I can't tell you which one is from an athlete or which one is from a person with longstanding hypertension. But if I pick it up, I can, because the stiffness of the heart actually responds differently to the different types of stresses. And that's why these stresses are pathological and induce disease. And so this idea that bad stress causes cardiovascular disease is sort of right in front of our face. If we look at the number of deaths by risk factor in the world, 3 of the top 5, high blood pressure, diabetes, and obesity, are the number 1 risk factors. So it's not surprising that the number 1 cause of death in the world is cardiovascular disease, outpacing cancer by about 2-fold. The same thing's true for us here in the United States. 1 in 4 deaths in the United States are due to cardiovascular disease. But sort of more interestingly, that isn't the complete picture. About half of US adults have hypertension, about a third have diabetes. And so even though this isn't cardiovascular death, this type of pathological remodeling is happening in the vast majority of adults in the world. And so as clinicians, we know hypertension, obesity, and diabetes often come together. And this now has sort of established to create a certain type of heart failure. So we know that this creates this HFpEF, or heart failure with preserved ejection fraction. And the characteristic of this heart failure is that the ventricular wall muscle becomes stiff. So while this muscle is able to contract normally and generate force to eject blood, the stiffness prevents the left ventricle from filling with blood. And of course, the amount of blood that's pumped out of the heart has to get refilled. And so the stiffer your heart is, so stiff hearts pump less blood. And this is because they can't fill, not because they can't squeeze. And this is something that my lab is really interested in understanding in terms of what are the molecular signatures of this stiffening. And so the stiffness of these cardiomyocytes, is the stiffness of a cell, is defined by your cell's skeleton, the same way our shape is defined by our skeleton. And these cytoskeletal filaments, sort of the bones of the cell, come in different flavors. And so actin, which forms the thick filament of your muscle, has been widely studied in muscle cells. So muscle people are going to be very familiar with this. And of course, is loaded entirely into the cardiomyocyte. And so this drives the contractile performance of the cell. So we want to soften these cells. We probably don't want to mess with actin because it's going to affect the contractility of the cell. Intermediate filaments, these floppy intermediate filaments, and microtubules have been somewhat less studied in cardiac and skeletal muscle. And so here's an immunofluorescence image taken from our lab of the microtubule network in the heart, which is this blue color and the orange, and the intermediate filaments, which are the purple color. And the primary intermediate filament in your heart is desmin. It forms these sort of end-to-end junctions between cells, as well as binds to the Z disc and aligns the contractile sarcomeres. But we had this question during my postdoc. Well, these stiff microtubules are running parallel to the contractile axis of the heart. What are they going to do when the heart cell contracts? And so this observation was made by Pat Robinson with ARIScan when I was in the Prosser lab. You could see here a cardiomyocyte being stimulated to contract, and the microtubule network is buckling at a high mode under like springs. And so what we think about this is this type of behavior right here, where energy is stored in these microtubules and they're returned during contraction. At least this was our original thought. And a back of the envelope calculation, what we determined was the amount of energy that's likely stored in these microtubules based on the number and stiffness is about 20% of the total force generated by the contracting cell. So enough to affect the contractile performance. And so I mentioned we had this access to human hearts. And so we isolated cardiomyocytes from healthy, on the left here, human cardiomyocytes versus failing human cardiomyocytes. And really across all etiologies of heart failure, we saw this densification of the microtubule network. And so at the time, it was really open in terms of determining, well, how are these microtubules impacting the stiffness of the heart, especially because they're densifying here in end-stage heart disease. And so the experiment that we did to figure this out, you can do this by stretching the cell or simply poking on the cell. This is an example using atomic force microscopy to poke on the cell. And we wanted to characterize the viscoelastic properties. And so the way that you do this is we measure the stiffness as a function of the speed with which we poke the cell. And you can see this positive slope here, which is indicative of the cell's viscoelasticity. So the non-failing cardiomyocytes exhibit this viscoelastic stiffness. And we found the failing cardiomyocytes were stiffer. In particular, the slope was greater, so they had this increase in viscoelasticity. So again, I showed that GIF or that image of Tigger bouncing like a spring. And so when we depolymerize the microtubule network, the drug that we use is colchicine. This is FDA approved for the treatment of gout. And this readily depolymerizes microtubules over about 90 minutes in cardiomyocytes. You can see that image shown here. And what we expected was that these curves would just shift straight down. That's not what we saw. So in fact, microtubule depolymerization did decrease the stiffness as we expected. But more importantly, or more interestingly, it decreased the slope of this relationship. And of course, there are more microtubules in the failing cardiomyocytes, so we saw a greater effect in the failing cardiomyocytes. But what this really indicated was that the microtubule contribution to the mechanics was viscoelastic more so than it was purely elastic like a spring. And so the next question was, what effect does this have on isolated cardiomyocyte function? And what we found first was that there was really no change in the calcium transient. So presumably the cardiomyocytes are generating the same amount of force with and without their microtubules. But when we looked at the cell shortening, we saw the purple is without microtubules with colchicine treatment. There was an increase in the shortening. It shortened faster, and the cells relaxed faster. So this discussion about diastole and relaxation is going to become important with this accelerated relaxation. And so this was consistent with the mechanical effect, but the feedback that we get, well, these cells aren't doing any work. They're not shortening under any load, where they'll shorten slower and potentially have difference. And so this is where it was really important for us to be able to turn to the system developed by IonOptix and Brad Palmer. He already showed the example of this. So our clips for the tissue are slightly different. This is a tissue slice from a rat shortening under load. We can have the length change in response to force. We could change the afterload, and that's shown here. But if we really want to assess the diastolic function related to how it would be in the clinic, we have to sort of think a little bit differently about what's going on during this phase of diastole. So I won't introduce the work loop because Brad already talked about this nicely. But basically the work is the area in the loop, as Brad said, pressure times volume. The one-dimensional analog of that is force times length. As you can see, pressure is force divided by cross-sectional area. Volume is length times cross-sectional area. You can cancel the areas out. We define the afterload and the preload in the work loop, and systole is the contractile phase, and diastole, diastolic dysfunction, occurs during this relaxation phase and the ventricular filling phase. And where stiffness really matters is at the end of this diastolic pressure-volume relationship. And so if you have diastolic dysfunction, the first way it's going to be assessed is through this transmitral Doppler. So this is echocardiography looking at blood flow into the left ventricle from the atria through the mitral valve, hence transmitral. And you can see here there are these two peaks of that velocity. There's this early filling phase, which is associated with myocardial relaxation. So this is what occurs with the original setting of the preload in the slice system. And then there's this late filling phase that occurs due to atrial systole. So when left atria contracts, it stretches out the left ventricle during the end of diastole here. And you can see that there is a proportional filling of the blood here that's about 8 to 40% of the total filling of the heart. And it's in this phase that the passive relaxed heart is stretched and its stiffness becomes important. And so if there's an increase in the stiffness, you would expect that this A peak is going to go down. And so our goal was to see if we could recapitulate this in the slice system. And one of the motivations for this is, of course, we can assess these PV loops in vivo, but these experiments are very difficult. They're also very hard to do clinically because you have to put a catheter into the left ventricle, a conductance catheter, to measure the pressure and the volume. A couple downsides to this is that it's very, very hard then to control heart rate, to control systemic pressure. And if we want to use drugs that haven't necessarily been clinically approved yet, or we don't know how they work to try and figure out their specific effect on the heart, this is the case with SGLT2 inhibitors, which have recently been approved for the treatment of diastolic performance, it's hard to isolate what happens for, say, changes in blood pressure that are going to reduce the afterload to direct effects on the heart. So these off-target effects, which are maybe potentially on-target effects, can actually impact the cardiac performance. So in this case, we're really motivated to adapt the cardiac slice system to really do diastole. And so this is how we did it. So this just shows the sort of protocol that Brad has talked about before, where we take a slice of or a chunk of cardiac tissue, we glue it down, we vibratome the slices, we align them along the fiber axis, put them into our clip system, hook them up to the rig where we can do these work loops. But we apply this stretch at the end of the diastolic phase. So this is sort of the unique part. And so this mimics atrial contraction and gives us an early filling phase where relaxation completes and there's a pause. And then we do a stretch before we go back through systole. So now we have the isovolumic relaxation phase, an early filling phase, and a late filling phase. And if we take the derivative of this length trace shown here, we can get what's effectively a velocity of expansion of the ventricle, which mimics this mitral Doppler result. And so to do this and to look at the effect of colchicine, we discovered very quickly, because it took 90 minutes to depolymerize the microtubule network, that if we used a typical Krebs-Hensel's buffer as used in a lot of these experiments, we had this continuous reduction in our total force. And so we moved to a media that was supplemented with physiological calcium. And again, we're continuously oxygenating this. And we could see that we got appreciable force generation, but more importantly, over 90 minutes, we didn't have a significant change in the total force associated with those isometric twitches at 1 hertz. This is the work of my graduate student, Emily Hancock. And so here's that DMSO trace, or the control, showing that over 90 minutes, we have a very stable contractile trace now in the DMEM with the right calcium. We depolymerized the microtubule network with colchicine. Before that, surrounded by this purple isometric twitch trace. And then with colchicine, we're a little bit surprised to see this. We generally saw a slight increase in the total contractile force and an acceleration in the relaxation. You wouldn't necessarily expect this from what I talked about in terms of the mechanics. And so now we're ready to look at the work loops. And so we applied the work loop with our end diastolic stretch. So you could see that stretch phase happening here. And after the 90-minute treatment with colchicine to depolymerize the microtubules, we saw this rightward shift in the work loop. And so the sort of metric for stroke volume, or how much blood is ejected during the contractile phase of the left ventricle, is the width of this work loop. So you could see, hopefully you could appreciate that this work loop increases. So the stroke length shown here, one of the parameters we can pull out, and colchicine improves the stroke length. Again, Brad had mentioned the area is the work. We see an improvement in the work, perhaps because the microtubules aren't acting viscously to dissipate energy in the slice, and that's shown here. And then the other parameter we were interested in is this end diastolic stiffness, this force length relationship. Interestingly, it doesn't change with colchicine the way perhaps it does in isolated cells. But that's because at the constant preload, we have this rightward shift in the work loop. And so if we look at the diastolic elastance, which is the diastolic and diastolic force divided by the total stroke length, we do in fact see that colchicine is shifting this down. And so we could take the derivative of the length trace to sort of look at our diastology and see how those different phases of the diastolic cycle are changing, specifically with the microtubule network. And just to remind you guys, so there's systole right here, then there's this isovolumic relaxation phase, and your two peaks for early and late filling associated with transmitral Doppler. The dark purple traces after colchicine. And I hope what you can appreciate from these average traces is that this isovolumic relaxation phase is shorter. We more quickly get into the early phase of diastolic filling that happens more and at a higher peak. So we have an increase in the E peak shown here. And interestingly, there's really no change in the late filling peak, and sort of nuance, but this is due to that rightward shift, we think, in the work loop. And so I think we really learned something about how the microtubules are able to affect diastolic performance that we wouldn't have necessarily inherently predicted from the original stiffness measurements and work that we did in isolated cells. And so on the work loop, we can extract the times associated with each phase from isovolumic relaxation, filling, contraction, and ventricular ejection. And so what's important about this is that it's this isovolumic contraction and relaxation phase that gets faster with colchicine treatment. There's really no change in the duration of the ejection phase. And the consequence that C and A, or A and C being faster, has is that there's more time available for diastole. And so the first symptom that patients with diastolic heart failure often have, or diastolic dysfunction often have, is exercise intolerance. When they raise their heart rate they basically can't keep up. There's an elevation in diastolic pressure. And so having this enhanced filling duration is probably going to be beneficial for the performance of patients that are suffering from exercise intolerance. So we're really excited about this. And just to give an analog, so this is a paper that came out of, or is on bioarchives from Lucy Carrier's group, looking at an HCM mouse model. So that's this red trace right here, treated with a microtubule deep tyrosinating agent, TTL, which is sort of has the same mechanical effect that colchicine would. And I hope what you can appreciate from these in vivo work loops is that, in fact, there is this rightward shift in the work loop here that mimics what we've seen in our data on the rats. And this is sort of evident here by this sort of enhanced and early filling and rightward shift. And, so the poll question that I wanted to ask everyone is, how interested are you guys in this type of diastology? Because I know we've added it onto the IonOptix system, and it may be something that could be sort of folded into the IonOptix software in the future. So with that, I just want to thank everyone for their attention. Obviously, thank Brad and my lab at UVM. This is a picture of everybody. And IonOptix for giving me the opportunity to share my work with you guys today. And you can find out more about our stuff on our website. Thank you so much.
Thanks, Brad and Matt, for wonderful sort of uses of the Slice platform and how you guys are developing the system to look at rat and other species primary slices. We're going to switch gears now a little bit. My name is Ben Lee. I'm one of the cardiology fellows and postdoc working in Ken Margulies' lab for the past couple of years, and so we're going to turn to looking at engineered heart tissues and how we've been able to adapt the Slice platform for looking at contractile mechanics in EHTs. And so this is going to be a brief talk just looking at brief background on EHTs, our tissue design, and some of the early contractile assessments that we're doing. So engineered heart tissues, just to introduce everybody, is, as methods to differentiate human iPS cells to cardiomyocytes and mature them have developed, many different methods have started to pop up with regards to just how to generate three-dimensional tissues. And these range from, spheroids and organoids all the way out to entire contractile and pressure generating chambers. And we'll focus sort of in this yellow portion or this yellow range that really balance the throughput of tissues and the tissue maturity, where thin strips are typically made and compacted against two anchors. Regardless, many of these EHT models are becoming more and more useful for a variety of different studies, including mechanism-focused studies, drug studies, organ or human on a chip models, and then for primary generation of tissues for muscularization. And so the one sort of underlying concept behind many of the engineered tissue models that exist is this concept of compaction. And in many such models, including models that our lab have used, as well as many others, what the tissues rely on and the cells rely on are a self-compaction within a gel. So you would mix iPS-derived cardiomyocytes, typically with fibroblasts and collagenous gel. And over the course, in this case of 7 days, some as few as 3 or 4 days, or as many as 28 days, will compact against two flexible anchors. And then these tissues can be further matured using electromechanical stimulation or maturation mediums that mimic the metabolic landscape in the developing heart. These bending sort of pillars or rods are incredibly convenient and elegant in the way that they can estimate oxytonic force, and I'll get into a little bit of what oxytonic means in a second. But by calibrating these flexible pillars against a needle, typically you can determine the force deflection relationship, and this allows you in many of these systems to measure force throughout the culture of engineered tissues, and learn a lot about their responses to things like drugs. However, the oxytonic forces don't mimic the cardiac work cycle that Brad was introducing earlier and Matt was talking about. So in pink here, you'll see a force length plot of what an oxytonic loaded tissue may look like. And that's in stark contrast to the 4 distinct phases of the work loop. And so, and again, in contrast also to the isometric contraction. And so because of these limitations to oxytonic forces, and oxytonic forces developed, it's difficult to sometimes do mechanism-focused mechanics or classical cardiac mechanics, and these might be necessary depending on the question that you're trying to study. And so what we've done is, and so, you know, if your tissues are large enough, you can adapt them to setups such as the IonOptix one, simply by piercing your tissue with hooks, or as with the slices, gluing them onto tissue adapters or tissue clips that then will place onto your hooks. We wanted to come up with a solution that was slightly more elegant, and in this way, we've generated our own tissue anchors here that inside a PDMS rubber well, we can cast our tissues, allow them to compact, and then lift the tissues entirely off of this well and do our phenotyping in a non-destructive fashion. And so when we make our tissues, they compact regularly over the course of 14 days. Here you see an engineered tissue just 1 day after casting, and over the course of the 2 weeks of culture, they sort of compact nicely around these two anchors that we've created. The tissues that we make are highly developed. So while at 7 days, the sarcomeric pattern is less developed, by 14 days of culture, we get highly aligned parallel sarcomeres in these elongated cells. And then the green here just shows the interspersion of fibroblasts that we have in our model system. And here it is hooked up onto the IonOptix Slice platform. We were able to lift the tissues off, and here are these two anchors and our engineered tissue in between. This is in comparison to living myocardial slice that we've been talking about all morning. And so what can we do once it's hooked up? Well, we can do anything that the system can do that you've seen a couple examples of today. First off, a simple isometric length tension analysis. And so in light purple here, a less stretched engineered heart tissue. And as we stretch the tissue, we get linear increases in passive force, as well as linear increases in active force. And then we can measure also the velocities that the tissue is making. It's hooked up to a myopacer, so we can pace the tissues at various frequencies. And here we see force frequency relationship in one of our engineered tissues, where we pace the tissues from a range of 1.5 all the way up to 3 hertz, or 180 beats per minute. And we see the effective rate of stimulation on metrics such as the passive force and the active force. And finally, we can also do work loops, just as you've seen a couple examples of today. Here's an example of 3 work loops generated by our engineered tissues, where again we see the 4 phases of the contractile work cycle, and what happens when we vary afterload from a lower afterload, higher work, to a higher afterload, and this in light green is in sort of a near isometric phase. And so, in summary, I think that we're really excited that the development of IPS-derived cardiomyocytes and engineered tissues have given us a method to cultivate cardiac tissues for both mechanistic and translational studies, and that oxytonic force estimates, while elegant and are able to provide measurements throughout the length of culture, provide a limited view of cardiac mechanics, and that transfer to dedicated force platforms enable more rigorous mechanical assessments, including ones that I've shown today: length, tension, force frequency, and work loop analyses. And so with that, I'd like to thank, obviously, the lab that I work in, Ken Margulies, as well as the other folks in the lab, Elise Corbin, who's in Delaware, who helped a lot with the conception of this project, and then the folks at Penn, who have been really supportive. Thanks.
Let's bring all of our speakers back. Brad, Matt, and Ben. Hi, thanks again for your presentations. Really clear and informative. We've got a lot of questions already rolling in, so are we ready?
Yeah, that sounds great.
Okay, our first question for this morning: how long can you keep cardiac slices in vitro? Is it long enough, such as days, to be able to transfect cardiomyocytes and therefore express a gene of interest before conducting experiments?
If you don't mind, I'll take this question. That's a great question. We've been able to keep a cardiac slice on the microscope while collecting data for up to 36 hours. But we can keep it in the incubator for weeks. And so and we do intend, and we actually have had some luck transfecting, but not the cardiac myocytes themselves. And I think that was just because we were using the wrong vector. But we have been able to transfect fibroblasts. And there are other groups, particularly the Dendorfer group in Germany, that's had tremendous luck with keeping slices in the incubator for weeks.
Fantastic. Anything else to add from the other two?
I might just add that there's some practical considerations to this too, in terms of how you keep the tissue, that it might need to be continuously oxygenated, that you might want to pace it, and some of these groups have developed strategies to work around this. So I think while it is doable, it could definitely be something that is being improved and is going to become more of a regular approach that's used.
Okay. Thank you both for your feedback. The next question, and I believe this was during your talk, Matt, what temperature were these studies performed? Is there any temperature dependence of the colch treatment?
So I can't speak to whether the colchicine treatment itself has a specific temperature-based kinetics. We use 10 micromolar of colchicine. These studies are done at room temperature. We've gotten an inline heater and are working on getting things up to 37. Microtubules are more stable at 37 degrees. And so these effects, if we were to predict, would actually probably be greater where the tissue is able to be paced faster and where there's an increase in the microtubule stability.
All right, thank you. Next question. What is the significance of measuring diastolic sarcomere length? probably any of you can jump in for this one.
I'm sorry, could you please say that one more time, Sydney?
Sure. What is the significance of measuring diastolic sarcomere length?
Well, if you have a measure of diastolic sarcomere length, you know where you are on the force length relationship of muscle. There's typically an ascending limb of the force length relationship, which is what's responsible for the Frank-Starling law of the heart. And so you get an increase in force production as you increase sarcomere length, but that's only at the short sarcomere lengths. If you were to go past about 2.2 microns in cardiac muscle, that typically levels off and you get no more increase. And then if you increase the sarcomere length even further, you'll get a reduction in developed force production. So the diastolic sarcomere length can be quite important because there's many different modes of contractility or force production depending on your sarcomere length.
Fantastic. Thank you. And then the next follow-up question is, can colchicine be used in HF?
I'm assuming HF means heart failure. So colchicine is FDA approved for the treatment of gout. The maximum concentration that a person can tolerate is about 24 times lower than what's needed or we think is needed to depolymerize cardiac microtubules. So it's unlikely. And you have microtubules everywhere in your body. So I think as generally, it's going to be important to develop more targeted therapeutics that specifically target cardiac microtubules or pathways by which microtubules are being stabilized in heart failure precisely.
Great answer. Thanks. Anything else to add from the others?
Yeah, just that the side effect profile of colchicine at higher doses just gets really untenable. And so that's going to limit its clinical use. Okay.
Thank you. All right, our next question. Can you record calcium transients with the myocardial, sorry, it moved on me. Here we go. Can you record calcium transients with the myocardial slices or the EHTs?
I think yes. Right?
Yes, we've been able to do it with FURA2. We can also get signals from FLUO4. You do have to be aware that you'll get significant motion artifact, and so it might be best to use a ratiometric dye such as FURA2.
Okay, great. This is a question for all three of you. Do you see considerable rundown during work loop experiments?
So I'll say that for the engineered tissues, we've had work loops running for 60 to 90 minutes and haven't seen considerable rundown. We do our experiments in sort of the full culture medium. And so that's inclusive of not only glucose, but fatty acids, serum, supplementation, and I think it's probably 1.8 millimolar calcium. So we don't see rundown in our EHTs over the course of about an hour, hour and a half.
Hey, Ben, do you need to oxygenate your EHTs?
So we actually haven't. They've been okay in 21% oxygen. That's how they're grown in the incubator anyways.
So
but it's something that we're looking at.
For us in the adult tissue, if you use this standard Krebs isolation type buffer, we do see rundown every 90 minutes. Also, if you shut off the oxygen, you see an increase in diastolic tension, and the slices do appear to get ischemic over a period of just minutes. So this is why we also switch to more of a culture media. And the reason that we currently don't have serum is just because it creates a foam in our bubbler. So for us, that's a little bit of a problem because we do need to oxygenate and have plenty of nutrients around for these things. So they're very metabolically active, but they'll run for a very, very long time with minimal rundown, which is the sort of other bit of the question.
Great. Thanks both for sharing your personal knowledge and experience. Okay, this question's directed for you, Ben. How similar is the generated force to real cardiac tissue? How is the calcium cycling?
Yeah, not similar. I mean, I think that this is one of the clear limitations of using engineered tissues as opposed to primary adult tissues. Is that the maturity of the engineered heart tissues is quite low compared to adult tissues. So the forces that we see are going to be less. That being said, you can still do comparative studies from engineered heart tissue to engineered heart tissue. And there's a whole field in the EHT world just focused on maturation of engineered heart tissues to get them to try to T-tubulate and look more adult-like. I think that there's still a little ways to go.
Okay, thanks. Let's see. Our next question. Can you comment on the strength and weakness of measurements of PV loop collected with the slice by comparing with those collected by the in vivo technique?
Well, I can tell you that, first of all, it's a lot easier especially if you're trying to measure pressure and volume in a mouse heart. That's extraordinarily difficult. And there's only a few laboratories in the world that really can do it well. On top of that, we can measure sarcomere length with a slice, which you cannot do in the whole heart. And so it's not that of course in vivo is certainly relevant and certainly pertinent. It's just that you don't have an experimental control over something like diastolic sarcomere length like you would have in the slice.
And so, I chime in on that too, in the sense that, and I think Ben has some to add. Certainly, you can't control as many parameters in vivo in terms of heart rate, preload and items like that. And then some of these drugs will have effects on other parameters. Well, it is really important to make these measurements in vivo. Sometimes teasing out the mechanism is best done in slices. And in addition to that, we're able to get multiple samples per heart from the slices, so we can increase the number of replicates that we're doing and look at sort of some of the differentiation there.
Yeah, I mean, the only thing I'll add is that while there's clear relevance to an in vivo system, I think that slices are convenient in the way that they're generated and we can use them, and offer distinct advantages over systems such as a Langendorf system, where you can also measure pressure volume, but that's a different ex vivo system that's complex to set up, whereas the slices are simple to use, and you get several slices per, for example, rat heart.
Okay, thanks all three of you. Great answers. Have you compared normal heart tissue to failing heart? So HFpEF versus HFrEF.
So that's heart failure with preserved ejection fraction versus heart failure with reduced ejection fraction. So I've done some stuff with heart failure with preserved ejection fraction. We're still analyzing the data, so I can't tell you too much in terms of what comes out of that. So that's my take on it. Have you done anything with these things, Ben?
Yeah, so so in the Margulies lab, we're regularly getting end stage HFrEF hearts and also donor non-failing hearts, and we're starting to do some of the comparative studies looking at how slices might differ in the advanced heart failure state relative to a non-failing state. It's rare for us to get HFpEF hearts. And then we can turn to animal models as well to start to do this, but we haven't done too much of that yet.
Okay, fantastic. Thanks for clarifying. We are almost out of time. I think we have time for one, maybe two more questions. So this next one is, do myocytes de-differentiate? Example, T-tubule loss and microtubule disarrangement when slices are kept in the incubator.
I'll say I'm not sure about that because we haven't looked at it and we aren't regularly keeping the slices right now, at least from my side.
Yeah, I think just from a theoretical standpoint, the moment you take a heart out, you need to be concerned with the the biomechanical, the electromechanical state that the tissue is in. And so things like oxygenating full media and keeping the slices happy in a biomimetic state may be the best way to sort of mitigate any remodeling that occurs once the heart's out of the body.
Okay, great. Let's get one last question in here for today. Have you compared function of left and right ventricle cardiac slices?
We haven't directly compared them in a sort of systematic sort of way, but we are able to generate slices from the right ventricle, at least from rats. From the mouse right ventricle, we found the right ventricle was already so thin we didn't bother making a slice. We just put up the entire right ventricle and it still generated work loops. So it's possible to make the comparison, we just haven't made it.
Okay, well, we got a lot more questions in, but we are out of time for today. So just letting the audience know, you can keep submitting your questions. We will be passing them on to the 3 speakers today. And I just want to let you all know that it was such a pleasure to have you with us. Really great presentations. It was, yeah, great to hear your research and your information.
Thank you for hosting us, Sydney.
In closing, we hope that you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by IonOptix, and I hope you all enjoy the rest of your day.